Thank you to the New Horizons Organizing Committee for inviting me to come speak today on the impact of Project Optimus on pediatric drug development. My name is Julie Bullock, and I'm currently the head of clinical pharmacology and translational medicine at Certara. My experience in drug development started 18 years ago at the FDA as a clinical pharmacologist, where in addition to reviewing adult development programs, I also reviewed a number of pediatric written requests and submissions for the therapeutic areas of hematology and oncology. Today, I will outline the goal of Project Optimus and go through some considerations regarding endpoints for dose selection for oncology. I'll then discuss the impact of Project Optimus on development timelines by comparing pre-Optimus development programs to a hypothetical future-looking Optimus development program. Lastly, we will discuss dose optimization in the context of pediatric drug development, including when extrapolation of optimal adult dose could be applied and when optimization in pediatrics would need to be considered. Project Optimus is an initiative from the Oncology Center of Excellence at the FDA to reform the dose optimization and dose selection paradigm in oncology drug development. While the Project Optimus initiative is focused on oncology drugs, it has direct applicability to drugs developed for rare diseases, which can struggle with dose selection and often lack dose and exposure response information to support dose justification for registrational studies. So while a lot of what I will discuss today will be centered around oncology, the overarching messages regarding endpoint considerations, impacts to development timelines, and applications to pediatrics are still applicable to other rare diseases. The goal of Project Optimus is to adequately characterize the exposure safety and exposure activity relationships to select dose for registrational trials and eventually approval. Historically, oncology assets have chosen the maximum tolerated dose based on safety, with little information to support the dose from a broader efficacy perspective, and establishing these therapeutic windows based on activity and toxicity is a critical part of development that is commonly seen in other therapeutic areas. By using non-clinical data, oncology assets can typically define target saturation and narrow a range of doses for further clinical evaluation, but more well-defined biomarkers, including response rates, tumor dynamics, or other clinical relevant endpoints, will need to be considered to support dose-response relationships. In this slide, I present two examples of a dose-response relationship. The one on the left is a typical expectation from cytotoxic or non-targeted drugs, where as we increase dose or exposure, we increase efficacy, but we also increase toxicity as well. The dose response on the right is more common for targeted drugs, and once the target is saturated, the efficacy typically plateaus. But the toxicity curve will continue to increase as we give more drug that is hitting off-target receptors. By evaluating efficacy at more than one dose, Project Optimus will further establish if we see commensurate gains in efficacy as we continue to increase dose or exposure, and if those efficacy gains are balanced by the toxicity seen at these higher exposures. In summary, the old way of identifying dose based on toxicity alone and taking that dose to registrational trials will need additional evidence to support the justification in the era of Project Optimus. In addition to evaluating additional dose levels for safety and efficacy, dose and exposure response relationships should be established and put into context with the dose intensity and adverse event time course. Toxicities past the first cycle DLT criteria must also be considered in this dose justification argument, and so should adverse events that affect patient quality of life, such as nausea, vomiting, diarrhea, and fatigue. Just like the rare disease indications, the endpoints we use to justify a dose in oncology can be varying. I find it easiest to bucket our endpoints into clinical benefit endpoints, surrogate endpoints, and what I'll call informative endpoints. Clinically meaningful endpoints relate to the outcomes which capture how a person feels, functions, or survives. The gold standard clinical benefit endpoint for oncology would be overall survival. But since clinically meaningful effect for the purposes of FDA is defined as how the patient feels, functions, or survives, quality of life is another endpoint that could be used, although rarely is leveraged for clinical benefit purposes alone for oncology. The next bucket is surrogate endpoints. These are endpoints that are reasonably likely to predict clinical benefit and are often used to support accelerated approval, as they are endpoints that can be collected earlier than a long-term endpoint like overall survival. However, some of these endpoints could also fall into the third bucket of clinical benefit endpoints, depending upon whether or not the surrogate is validated for the indication. The last bucket is informative endpoints. These include biomarkers, which do not directly relate to how a patient feels, functions, or survives, but instead are objectively measured indicators of biological or pharmacological response to a treatment intervention. Biomarkers may include pharmacokinetics, circulating tumor DNA, blood tests, tissue samples, imaging results, or physiological measures which are used for diagnostic, prognostic, or monitoring, including safety purposes. These informative endpoints assist in understanding the characteristics of your asset, but alone are not sufficient to support clinical decision-making for approval process. Their utility for dose optimization, however, is important, and these endpoints should be used to narrow the range of doses for further testing in both adult and pediatric clinical trials. Next, I will discuss how the impact of Project Optimus is on development timelines. An example of a historical oncology development timeline and when pediatric drug development would be discussed is presented here. We all start with first-in-human multiple ascending dose designs, which provide information on safety, PK/PD, and activity. And then we typically move to expansion, where 20 or more patients are treated at a single dose level. After expansion, most oncology assets are discussing registrational studies with the FDA, so this would trigger the initial pediatric study plan submission. In this framework, the pediatric development is being discussed often before we understand the efficacy of the asset in the adult population. Any understanding that we have at this time is usually quite limited, as it's coming from the first-in-human study and the expansion cohorts, often at a single dose level. Here I overlaid Project Optimus era development timelines on top of the historical timelines. And you can see how the dose-finding exercise delays the end of phase 1 meeting and the discussion of registrational trials. Therefore, the initial pediatric submission plan will be also delayed. However, by the time the IPSP is discussed with the agency, a better understanding of the drug's risk-benefit in adults will be known, and this should mitigate some uncertainty in the pediatric oncology development community with regards to doses needed to target efficacy, since the IPSP submission would be discussed typically after dose finding has initiated or is potentially completed. I'll pause here to mention that the option for the Type F meeting allows sponsors to discuss strategy and contents for the IPSP prior to submission. These meetings are intended to provide an opportunity to discuss the agency's current thinking about the relevance of a specific target and the expectations for early assessments in the pediatric population, unless justification for a waiver or deferral of a pediatric study can be provided. These meetings can be held in between the end of phase 1 and the end of phase 2 meetings to discuss the adult indication, and would be best supported once the approach to dose-finding adults has been agreed upon with the FDA, or we have results from the dose-finding exercise. Next, I'll discuss how Project Optimus and dose optimization is being applied to pediatric development. For indications where efficacy from adults can be extrapolated, the pediatric studies should leverage the data collected to support the adult dose justification, and the pediatric studies should be able to target PK matching to the optimal dose. This is probably the most straightforward case, and while safety will also need to be established for pediatrics, the burden of efficacy may be lessened if the adult dose justification is sound. For indications where efficacy extrapolation is not possible, the prior understanding of the target and efficacy modulation, along with the safety across the dose range from adults, should be critical to shorten the dose-finding exercise in pediatrics. But since efficacy cannot be extrapolated, the dose that provides the optimal risk-benefit in children will need to be established. Ideally, in this scenario, comparison of adult and pediatric preclinical xenograft models should be able to assist to generalize relevant dose and target modulation from adults to pediatric, even if the indications are different. Last, for development programs that will focus on pediatric indications, the dose optimization exercise will likely need to be conducted in pediatrics, since no dose finding would have been done in adults. And the approach should provide the integrated analyses as suggested and discussed earlier by Project Optimus. This will ascertain that the dose taken to pediatrics provides for the appropriate risk-benefit in these indications. Thank you for your attention, and I look forward to your questions at the panel discussion.